Today we're going to go over Mendel's other principles, okay? They're a little bit complex, but in the end, you could understand them because you got a good foundation for meiosis. So when meiosis happens, we're going to produce gamete cells, which are sex cells. Mothers produce gamete cells, fathers produce gamete cells. What Mendel realized is that during the formation of those gametes, alleles separate so that each sex cell carries one allele. In other words, the mom has two, she's going to give one gamete one allele and the other gamete another allele. Make sense? Okay, so here's a picture for you. You've got two parents here. What can you tell me about those parents? Yes? They're homozygous. What kind of cross is that?
Purebred.
A purebred cross. You've got two homozygous parents, that's a purebred cross. This is showing you meiosis. And in meiosis, we produce gametes. So here's my mom right here, and here's my dad. Mom is going to produce a gamete no matter what with a capital S, because that's all she has to give. Dad is going to produce a gamete with a little s, because that's all he has to give. If I take those two gametes, and fertilization occurs, which is right here, we end up with my F1 generation. What else can we call that? A hybrid, good. And we know my hybrid is always going to be heterozygous, correct? And here's the heterozygous hybrid. Now, this hybrid produces different types of alleles. So this hybrid is going to go through meiosis when puberty occurs, and they're going to start to produce gametes, okay? These gametes can be capital S and little s. Now they could be two different pairs, or two different gametes, as opposed to this one only had one choice. So I'm going to go to the next one so you can see what. So this one goes a step further. It starts with the same thing, my purebreds. They produce only one type of gamete because that's all they can make. And then you've got your F1s. And here's one F1 and here's another. That's what it means by self, let's say. Okay? Two different F1s.
Is that the result of the
That's the result of a parent.
So both
Yes. This is the possible gamete combinations for this offspring, and this is the possible gamete formation for the other offspring, which is the same because they're both heterozygous. What shows to be different is when they cross. So this is a heterozygous with a heterozygous, which we know is called what kind of cross? F1 cross. And the offspring is called F2. This is my offspring. So my offspring, who could tell me what is the genotype ratios for my offspring? Actually, I'm going to go a little bit simpler. Who could tell me what is the genotype of this offspring? Capital S, capital S. If you didn't remember that today, somewhere in your notes, draw a star of this caliber and write genotype always means letters. Always. So you do not forget that. Okay? So the genotype of this offspring is capital S, capital S. What about this offspring?
uppercase, lowercase.
Uppercase, lower. This one is the same, and this one is lower, lower. Now, who could tell me the genotype ratios for this offspring?
25% capital S, 25% lowercase s, and 50% capital S, lowercase s.
Good. 25 capital S, 25 little s, and 50% capital S, little s.
That F1 cross creates the F2 generation.
Correct. F1 cross equals F2 generation.
And it can only be an F1.
What is the phenotype ratio for this cross, for the F2 offspring?
75% round and then 25%
Okay, so he said round. Look at the letters. We know that we choose the capital letter for the dominant trait. And I haven't even told you what it is. But just looking at the picture, try to come up with something that makes a little bit more sense. Yes? Okay. How much percent smooth? 75%. And 25%? Rough or wrinkled. Good. The opposite of smooth. And do we really know that? No, but that makes logical sense. Because the capital S shows the dominant trait. All right, so we move on. Then he also developed what is called the principle of independent assortment. So up until now, we've only looked at a pair of alleles. We haven't looked at more than one pair together, but realistically, if your cells have 46 chromosomes, or 23 pairs, and on those chromosomes you have all these genes, alleles are constantly being separated, and they do this independently of each other. In other words, it doesn't matter where one allele goes, any allele can follow. There's no set order. And I'm going to show you a couple of different diagrams, so hopefully this makes sense, and then I'll show you a video. But when you have a parent here, right here, here are my parents, we know what about those parents? They're purebred. For how many pairs of alleles? 2. You got alleles for round and wrinkled, and you got alleles for yellow and green. When gametes are produced, you're going to get this combination from a homozygous parent. Capital R, capital Y, or little r, little y. But if we take over here, and we move across to here, here's that gamete. Capital R, little r, capital Y, little y, heterozygous. That's my F1. Look how many different gametes that can produce. You can have a capital, capital, you can have little, little, or you can have capital, little. It doesn't matter. There's no order that says because you're dominant, you must travel with the dominant. Because you're recessive, you must travel with the recessive. Each allele is independent of their surrounding alleles. There's no set order. So now you take that and look how many different combinations you have if you do a Punnett square. This is what we were talking about yesterday. If you take 2 alleles and you perform a Punnett square, you're going to have 16 possibilities of combinations, not 4. Yes. So sex-linked traits are basically traits that are going to be found on the X or Y, and 99% of the time they're found on the X. Okay? X and Y are your 22 pair chromosome, which determines the sex. If you're XX, you're what? Female. If you're XY, you're a male. Now, we're going to write sex-linked traits as the following. The same way I taught you how to write dominant and recessive traits, well, now they're dominant and recessive, but they're on an X or a Y chromosome. And we're going to focus only on X, okay? If I tell you that there's a genetic disorder that is sex-linked and is recessive, then we're going to use N for normal, for the dominant trait, and little n for the disorder. To the left, what are all my possible combinations there? Female or male? Female. XX is female. So to the right, we've got my male. Okay? Now, looking closely, a female that has X little n, what can you tell me about her? She has the disorder. A female that has X little n, X capital N. Good. She's a carrier. Does she show it? No. She doesn't have it, she's normal, but she carries it. This is where you hear like specific people that know they carry a genetic disorder might have testing done before they have offspring, just to make sure, okay? Because if you know you're a carrier, you're gonna consider really, really well, and you're gonna learn this in your genetic disorders, whether you wanna give birth knowing there's a 50% chance your offspring is gonna have the same thing. Yes? Normal. So men, you have it a little bit worse than we do. Because you don't have two X's, okay? In this case, you're normal. But when you carry one recessive allele, you're doomed.
That's inspiring.
You don't need two. You get one and it's over. So sex-linked traits for males, there's a stronger possibility of having it. Because 99%, now I'm not saying every sex-linked trait is a disorder because some are not. And I'm gonna tell you right now, guess what one sex-linked trait is? Who gives you your genetic disorder for baldness? What? Your mother. So today, look at your mother when you get home. If your mother has very, very thin hair, not like this, welcome to the hair club for men, because